This expansion microscopy protocol enables the investigator to visualize glomerular proteins with nanoscale resolution on a conventional microscope.
The main advantage of expansion microscopy is that it is accessible to a large research community.
Begin by making spacers for the gelation chamber. Cut number 1 and number 1.5 glass cover slips into 5 mm stripes using a diamond knife. Place a glass slide into a staining chamber and position the number 1.5 cover slip stripes on the glass slide so that they form a 2.5 cm square. Pipette a droplet of double distilled water into the corners of the square to adhere the glass cover slip stripes to each other and to the glass slide. Wait for approximately 20 minutes so that the number 1.5 cover slips are stably attached. Pipette a droplet of water on each number 1.5 cover slip stripe. Place 4 number 1 glass cover slip stripes onto the number 1.5 cover slips. For the gelation chamber lid, wrap a cover glass with paraffin film, avoiding any folds or dirt on the film. For anchoring treatment, remove PBS from the immune-stained cells and add 250 microliters of anchoring buffer per well directly onto the glass cover slip. Incubate the plate for 3 hours at room temperature. Keep the substrate in the dark using a box. After the incubation, remove the anchoring buffer and wash the cover slip once with 1.5 milliliters of PBS per well. To stain actin fibers, thaw an EXM-compatible phalloidin solution and incubate it for